So my protein that I have been given is the KRAS protein, and the mutation that I will be looking at is the G12C mutation, in which glycine at position 12 is replaced by cysteine. So the normal function of KRAS is acting as a relay molecule, so it's relaying signals from outside of the cell to the cell's nucleus, and it's controlling how the cell knows when to grow, when to divide, when to mature, and how to go about its specific functions in order for that cell to survive. So it's normally found in human and animal cells, mostly human, and it's typically found in the plasma membrane, the cytosol, the mitochondrion, as well as the nucleus, and it's actually created by the KRAS gene, and the KRAS gene is on chromosome 12 in each of the cells of the human and the animal. So the mutation is actually making the protein more active because the KRAS protein is being constantly turned on, not getting that ability to turn off, and it's causing that cell to proliferate uncontrollably. And the mutation's protein structure is affecting its function by forcing it to increase the strength of the signals, so allowing that cell to grow uncontrollably and have those cells growing uncontrollably. And this protein's change in structure is happening in that amino acid sequence, in which the amino acid glycine at position 12 in the amino acid sequence is replaced by the amino acid cysteine. So this change in structure affects the mutated protein's function in only causing harm by it amplifying those signal pathways due to KRAS being loved at its active state by this mutated cysteine, and that can lead to these cells having oncogenesis and creating these cancerous cells. So the main functional groups present in the glycine is the carboxyl group, the amine group, and one side chain of only one hydrogen. So glycine is actually hydrophobic as a result, and it's actually the smallest amino acid out of all of them. So glycine is an inactive hydrophobic molecule, and because of that, it repels water. However, due to it having an allopathic side chain, it is able to either completely repel water or have a slight solubility for water, which leaves room for there to be a hydrogen bond, if anything, and that's the only bond that it can really handle. So in contrast, we have the cysteine, which is that mutated amino acid chain, and it can have those main functional groups of a carboxylic acid, an amino, and a sulfhydryl group. So the cysteine is polar, but it is not charged, and it is obviously larger than our glycine because our glycine is our smallest amino acid, as I said before. So with cysteine, we can actually have a disulfide bond because that sulfhydryl group is actually able to oxidize into that disulfide bond, and so its polarity also that it has is also able to allow it to make hydrogen bonds. And so, like I said before, also glycine is able to create those hydrogen bonds. The G12C mutation affects the protein's function by amplifying the protein's function of having that signaling for that cell growth and division, and it's happening uncontrollably due to it not being able to turn off. And so we have all of these cells going crazy everywhere, and instead of them being normal, they're cancerous, causing all types of issues throughout the body. And this mutation also affects the cell's function by causing the normal cell, as I said before, to possibly become cancerous and continue to grow and divide that way and create more and more cancerous cells. The mutation also affects the organism by limiting that organism's bodily system's abilities to do their jobs and keep that organism just surviving at its best potential. That's an example of lung cancer. Lung cancer is a very common cause of this G12C mutation, and as a result, the lungs can have many issues with breathing, and we can get a lot of heart problems as well. So it's really limiting the quality of life for that organism and limiting its body system's abilities to function normally. So now I'm going to go into talking about my model and the limitations to it. Here are my models for the mutated KRAS and the unmutated KRAS. I have the normal KRAS represented here, and we have our glycine represented here at the 12th position in the amino acid sequence, along with the GTP here. And then over here we have our mutated KRAS with the glycine being substituted with the cysteine here at position 12 in the amino acid sequence, along with our GTP again. As you can see, there's a difference in their size because, as mentioned earlier, the cysteine is larger than the glycine, being the smallest amino acid. And there are a few limitations to these models, and the first one being that the little less than 200 amino acids represented on the KRAS are missing on both of these models besides the glycine and the cysteine. The next one is that the magnesium ion that helps catalyze the hydrolysis from GTP becoming GDP to turn the KRAS off on the normal KRAS protein right here is missing. And lastly, the structures for the G12 here on the normal model and the C12 here on the mutated model are unrealistic in their structures.